All right, this video is for the biochemical test lab, and I'm going to be demonstrating the motility test and the TSI slant test. So I'll do motility first. The first thing you want to do would be to sterilize your bench before you start and put all your equipment down. And then once that's done and everything's set up, you would take your inoculating needle. Both of these tests actually use needles. You take your inoculating needle and you'd sterilize it for 10 seconds and then let it cool before you pick up your source culture and remove the cap with your pinky on this hand. And then you would sterilize the top of the tube. And because it's a slant culture, you don't have to mix it. It'll just be a matter of sterilizing the top of the tube and then taking your needle and grabbing a little bit. And then you would sterilize the top of the tube again and replace the cap and put the culture back in the tube rack before you pick up your motility deep. And you would do the same thing with the tube where you pick it up, remove the cap with your pinky, sterilize the top, and then you want to just stab down into it. Let's see if I can kind of draw a line here, about 3/4 of the way down. And you want to make sure that when you do it, you follow the same line in and the same line out, because the whole point of the culture is to see if the bacteria move. And if you don't follow the same line, then it's a lot harder to tell if they moved or not. So you'd inoculate it like that, being careful to follow the same line 3/4 of the way down. And then you would sterilize the top of the tube again, and you would place it in the incubator for 24 to 48 hours at 37 degrees Celsius. And then a positive result would be if anything besides the line is red. So if the bacteria spread out from the line, then you know that they can move. And that's it. And then for the TSI slant, it's pretty similar. Again, before you start, you would sterilize the bench, and then you'd start by sterilizing your inoculating needle. Again, it's a needle that you use for this one. So 10 seconds and let it cool. Take your source culture, take the cap off with your pinky again, and sterilize the top of it before you go in and grab a little bit. Again, just at the tip of the needle. You would remove your needle, sterilize the top of the tube again, and then replace the cap. And then you can put that back in the tube rack before you pick up your TSI slant. And with this one, it's pretty similar, except for that it's a slant instead of a deep culture. So you actually want to stab down into the butt of the deep. So you go down almost all the way to the bottom here. Let's see if I can get this to right. So you pretty much want to go all the way down because you need to see how the bacteria behave at the bottom of the culture, or at the bottom of the slant. And then once you withdraw the needle, again, being careful to follow the same line. On the slant portion of it, you just want to make one line up the slant. Like, hold on, let me see if I can get this thing to, just one line up the slant so that there's some at the surface as well. And then you would sterilize the top of the tube, and then replace the cap, put it back in the tube rack for a second so you have time to sterilize your needle before you put it down. So you'd sterilize this for 10 seconds to make sure that everything is dead before you set it on the bench. And then you can take your slant and put it in the incubator, again, 37 degrees Celsius for 24 to 48 hours. And both these tests don't really have any reagents with them. The slant itself and the motility culture do all the work for you. So that's it. And then I guess when you're done, of course, you would sterilize the bench again.